Move over to the second talk today, which is from Richard. Richard, are you there?
Yep.
Right.
You hear me?
Just maybe, yes, we can do. So this is Professor Richard Stanton from Cardiff University, who's going to talk to us about SARS-CoV-2 and ADCC.
Okay. Sorry?
Yeah, I was just saying that I was just confirming we can see your talk. So
excellent.
And hear you okay. So.
Okay, so yeah, I'm going to talk about some work we've been doing on SARS-CoV-2 and ADCC, starting off with some published data to sort of set the scene, and then going into some unpublished stuff. So we all, when we think about antibodies, we all know about neutralizing antibodies. These are antibodies that bind the virion, prevent it infecting your cells, clearly very important. However, if the virus is able to breach this barrier and get inside your cells, obviously these antibodies can't access it, and so they become a bit less effective. So at this point, what you need are antibodies that, rather than binding the virion, bind the infected cell. They're going to recruit things like natural killer cells to come along and carry out activities such as antibody-dependent cellular cytotoxicity, or ADCC, where the NK cell recognizes the antibody-bound target, kills it off, and controls the infection. And so these two antibody activities are really very complementary. This is an activity which targets the cell-free virion. This is an antibody which targets the cell-associated virion. Neutralizing antibodies have been a huge focus of many labs. Antibodies that mediate ADCC get a little less love, but this is where our interest lies. And so we've been characterizing this response in the context of SARS-CoV-2. And so the first thing we asked was simply, does infection induce antibodies that can drive ADCC? So what we've done here is taken some A549 ACE2 cells, infected them with SARS-CoV-2, and incubated them for about 5 hours with some serum from someone who is seronegative or seropositive, along with NK cells. And in any graph where I show percentage CD107A, you can basically take this as a marker of activation. Bigger numbers are better. If the numbers are bigger, your NK cells are more activated, and therefore they're going to be killing your infected cells better. And so you can see here that if we have the seropositive serum and we compare it to the seronegative serum, there's a massive jump in the ability of the NK cells to recognize and respond to the infected cell. And so this jump is the ADCC in action. So clearly, infection does induce ADCC. We want to ask the question, which antigens on the infected cell surface are driving this response? And so we used a quantitative proteomics-based plasma membrane profiling technique to identify all cell surface antigens. And if we focus here on just the viral antigens, we had 4 antigens there: nucleocapsid, spike, membrane, and ORF3A. To see whether these antigens could drive ADCC, we transfected cells with expression plasmids, along with various negative controls. And you can see that all 4, ORF3A here, membrane here, nucleocapsid here, and spike here, all 4 were capable of increasing the activation of our NK cells in the presence of seropositive serum. So they can clearly drive ADCC in the context of transfection, but of course what matters is what's happening during infection. And so we started off our analysis looking at spike, because we had a whole panel of antibodies to do this with. These antibodies target all different domains of spike, from the NTD to the RBD to S1, S2, etc. And they all bind the infected cell surface really nicely. Spike is expressed well on the cell surface. These antibodies bind it very effectively. And so it was a bit of a surprise when we then did our functional assays and found that actually none of these monoclonal antibodies were very good at doing ADCC. For comparison, here is a serum from someone who's been naturally infected. These are all the different monoclonal antibodies. Most of them do nothing at all. A few of them have some weak responses, but they're clearly nothing like what you get from a serum following natural infection. That was a bit of a surprise. We thought spike would be a good ADCC antigen. And so we wondered if it was just that we didn't have the right antibodies in our panel. Maybe the spike antibodies that drive ADCC are rare. And so we took an alternative approach, and rather than adding in monoclonals, we depleted spike antibodies from polyclonal serum. And don't worry too much about the scales here. They aren't comparable between experiments, but within each experiment, each paired line here is a donor before and after depletion. Bigger numbers, better ADCC. And so you can see if we have our wild type serum and we deplete spike antibodies from it, we see no change really in the magnitude of the ADCC response. If we start going to the other antigens that we're interested in, though, we do start seeing responses. And so here we've depleted nucleocapsid specific antibodies. And now we do start seeing a drop-off in the magnitude of the ADCC response. It's not a massive drop. There's still quite a lot of residual activity, but clearly nucleic acid antibodies do contribute. When it came to membrane and OR3A, we didn't have reagents to deplete antibodies against those proteins, and we couldn't delete membrane from the virus genome because it's essential. But we were able to delete OR3A from the virus genome. And if we look at ADCC activity against a wild-type virus or a virus that lacks OR3A, we now see a really massive drop in the ADCC activity. And this says that OR3A certainly is a major contributor to ADCC activity in the context of infection, possibly with some contribution from membrane, certainly some contribution from nucleic acid antibodies, but not much contribution from spike-specific antibodies. And so the obvious question is, that follows on from that, is what happens if you only have spike antibodies because you've been vaccinated with a spike subunit vaccine? And the answer is you can get ADCC. Most of our donors who've been vaccinated don't really show much ADCC at all. This is our best vaccine we've ever found. You can see there is some ADCC going on here, but it's far, far inferior to what we see with someone who's been naturally infected. The big difference, of course, here is this ADCC is totally spike dependent, so we can deplete the spike antibodies and the activity disappears. Again, that's very different to natural infection where you deplete the spike antibodies and nothing happens. And you can see that summed across multiple donors here. It's still on a log scale. Again, bigger numbers are better. You can see people who've had natural infection mount good ADCC responses, but those who've been vaccinated mount very poor responses. And it's not helped by a second vaccine dose either. This isn't because they aren't responding to the vaccine, because if we look at neutralization, they give, certainly after their second vaccine, they have very good neutralizing antibody responses. So at least as far as vaccination is concerned, there's a complete disconnect between the neutralizing response and the ADCC response, with vaccination inducing good neutralizing antibodies, but poor ADCC responses. And so what we really wanted to do at this point was say, well, if spike's not a great vaccine antigen for ADCC responses, what about some of the other targets we've identified? And we were able to start doing this with nucleic acid. It's the one antigen we've been able to do so far. I've just got a reminder here to say to myself, give a quick plug for preprints here, because the way this paper worked was we published our paper saying that things like nucleic acid are ADCC targets. And then a preprint went up saying that these people had vaccinated animals with a nucleic acid antibody and found it protected them. And so I emailed them and said, great study, that's exactly what we wanted to do. Have you looked at ADCC? And they replied and said, we haven't, but the reviewers have just come back from our paper and they want to know how it works. And so we ended up working together and publishing a joint paper. And so the vaccine strategy here was to vaccinate the mice with an adenovirus expressing nucleic acid followed by a N protein. If you do this, you get nice antibody responses in ELISA to nucleic acid, and you can see the nucleic acid and B cells in the memory pool here. In terms of functionality, these antibodies do nothing as regards neutralization. This is an assay here looking at the neutralization of our N-specific sera. For comparison, here's some spike-specific sera neutralizing very nicely. So N antibodies don't neutralize in the slightest. What they do though, is bind to the infected cell surface. So here's some flow data. These are our control animals. These are the animals that have been vaccinated with our nucleic acid vaccine, and you can see the antibodies bind to the cell surface very nicely. Although it has to be said, not to the extent that a nucleic acid monoclonal antibody did. And so we went on to do an in vitro ADCC assay with these serums. Here are our control serums. You can see the serums, the N-specific serums inducing ADCC activity. And again, this anti-N monoclonal, which bound better to the infected cell surface, gave much, much stronger ADCC responses. And of course, this monoclonal and even the polyclonal data as well, with the against the infected cells here, is in sharp contrast to what we saw with the spike monoclonals and polyclonals, where they were very weak ADCC inducers. These are much more potent. And so having shown in vitro evidence of efficacy, we went on to do the challenge experiments to make sure we're looking at just antibodies rather than T-cell induction things. The mice that had been vaccinated were bled and the serum was reinfused into naive mice prior to challenge. And if you do this and you look at titers of virus in the lung 5 days later, you can see that if you have N-specific sera, then you get a measure of protection, but the N monoclonal antibody which bound to the cell surface better and gave better in vitro ADCC, also gave superior efficacy in vivo. And so that's a really nice example, I think, of where antibodies that don't do any neutralizing activity but do ADCC can provide protection in animal models. Clearly, there's room for improvement in the vaccine strategy. I think these antibodies, we had high antibody levels, but their ability to bind to the infected cell surface wasn't as good as we might have wanted. And so that there is room for improvement in terms of inducing the right types of nucleic acid antibodies. But clearly, the monoclonal demonstrates the power of this approach. And so to summarize the published stuff, spike is a poor ADCC antigen when it comes to both monoclonal antibodies and vaccination. Into polyclonal serum, it's driven much more strongly by things like OR3A, membrane, and nucleic acid. So far, we've been able to look at one of these, nucleic acid, in animal models, and ADCC promoting N-specific antibodies are effective in animal models, even in the absence of neutralization. But of course, up to this point, we've really been looking at our human cohorts in terms of people who've been vaccinated or people who've been infected. Over the last year or so, those two groups have merged, and now the vast majority of people have been both infected and vaccinated, having what's termed hybrid immunity. And so we thought it would be interesting to see what goes on during hybrid immunity as well. So far, we've only done this in one particular setting for practical reasons. The setting where we've done it is in people who've had a very, very mild, almost asymptomatic initial infection. The reason for doing that is that if you've had a symptomatic or a severe infection with SARS-CoV-2, the ADCC antibodies that you generate against things like OR3A are so good that vaccination on top of that doesn't really have any impact. You can't see any effect of vaccination. So we focused on people who had asymptomatic or very mild disease. They therefore had very mild OR3A antibodies, for example, driving their ADCC. And we also waited for a while after their infections let these wane a little bit. And you can see that the response that's left here. This is the pre-vaccine response, and you can see it dilutes out quite readily. However, when we then vaccinate, we see a boost in ADCC responses from vaccine 1 and vaccine 2. And so if we look at this dilution here, 1 in 256 dilution, we've diluted out our OR3A and membrane and nucleic acid antibodies. They're no longer contributing. The only thing we've got left are our spike antibodies from vaccination, and therefore this is spike-dependent ADCC. So it is possible to get spike-dependent ADCC. It's worth pointing out that this response remains inferior to what you get during infection. So here we've got someone who's been infected, someone who's got hybrid immunity, and someone who's been vaccinated. And so you can see these really, really weak responses from vaccination, the really, really strong responses from infection, and the sort of intermediate response from hybrid immunity. But nevertheless, we currently have spike-based vaccines, and if we can learn how to induce superior ADCC responses from spike-based vaccine, that's clearly going to be a good thing. We've compared neutralization and ADCC in these cohorts. You can see in both cohorts, both the vaccinees and the infected people, there is a correlation between neutralization and ADCC. In the vaccinees, these are the hybrid cohort up here. These are the people who have just been vaccinated alone. But you can see the relationship between these two parameters is quite different in these two cohorts, in that for a given level of neutralization, you get much better ADCC in the infected cohort than you do in the vaccinee cohort. And that really goes back to the original point that these donors here have a wide antibodies targeting a wide range of antigens that are superior in inducing ADCC compared to these. But nevertheless, the real question was why are the hybrid cohort displaying better ADCC than the vaccine-only cohort? We did wonder if it was purely about IgG abundance. If you have hybrid immunity, you have higher levels of IgG. Two things argue against that. One is this graph from earlier on, where I showed vaccine 1 and vaccine 2 responses. There's a big boost in the amount of IgG after your second vaccine, but there is no boost in the maximal ADCC response. You can dilute it out further, but the response is not any higher. And the second thing we did was to compare directly levels of IgG with ADCC activity. And so you can see that there is a relationship between levels of IgG and ADCC activity in both the hybrid and the vaccinee cohort. But again, that relationship is very different. For a given level of IgG, you get superior ADCC in the hybrid cohort compared to the vaccine cohort. And so that really says it's the quality of the immune response that makes the difference here, not the quantity. To begin to dissect what was going on, we started looking at the where antibodies were binding across spike in these different cohorts. And so to start with, we just looked very crudely at S1 versus S2, the S1 domain being containing both the N-terminal domain and the receptor binding domain, and the S2 region being the membrane proximal region. And so if we look at the relative levels of antibodies targeting these two domains, you can see that following infection, the response is biased towards the S2 domain. If you then vaccinate on top of that, you really boost the S1 response dramatically, and you get just a small boost in the S2 response. And that's not really changed by your second dose. If you've just been vaccinated and never infected, then you're biased towards your S1 responses. S1 responses are higher than S2, and your second vaccine boosts this further. So your S1 responses become boosted very highly, your S2 responses only a little bit. And so the overall picture that comes out of this is that infection favors S2 responses, vaccination favors S1 responses. And so the only scenario where you get both good responses against both domains is if you have hybrid immunity. And so to see if this was the reason for the difference in ADCC responses between the hybrid and the other two cohorts, we went on to take serums to deplete them of antibodies targeting various domains of spike, and then to use these serums in an ADCC assay against live virus. It's a complicated graph. I'll try and walk you through it. Basically, we diluted these serums to the point where we've diluted out the O through A membrane nucleocapsid antibodies. And so we're looking purely at spike-dependent responses here. In blue is our starting serum. If we deplete all spike antibodies in the red, we lose our ADCC completely. If we deplete antibodies against RBD in the orange or NTD in the purple, then we don't really see much of an impact. If we, however, delete all of the S2 antibodies, we do see a reduction in ADCC. And similarly, if we delete all the S1 antibodies, we see a reduction. But neither of these depletions takes us down to the level of complete depletion. And so that says that really what's happening here is you need both S1 and S2 responses if you want to get ADCC against spike. One thing that was slightly surprising here is that we didn't see anything when we depleted NTD or RBD alone, but when we depleted the S1 domain antibodies where these domains reside, we did see an effect. So to find out what was going on, we took a different donor, did the same thing again, but as well as separate NTD and RBD depletions, we did a double depletion of both separate, both together. Looking at this donor, again, blue is the starting serum. This time depleting NTD alone or RBD alone had a small impact on reducing ADCC. Depleting the S2 domain, again, had quite a larger impact, and depleting either NTD and RBD together or all S1 antibodies also had a partial impact. And so the conclusion, or I should say, actually, there's quite a lot to get your head around. I've got a summary graph here. Here we're looking at multiple donors, and we're showing how much ADCC remains if you deplete antibodies targeting each of these domains. And so just to go over it again, if you deplete NTD or RBD, you don't see much of an impact on their own, but if you deplete both, you do see a partial impact. And that really matches what you see if you deplete all of the S1 or all of the S2 antibodies. And so what this says overall is that if you want good ADCC against spike, you have to have a really polyclonal response. You have to have a response that absolutely decorates that spike protein from top to bottom, all over it, to really get good ADCC responses. And that probably reflects the fact that it's not a great ADCC antigen. You need all these antibodies all over it to get good responses to activate those NK cells nicely. But we did wonder whether the fact that it was such a polyclonal response would make it more resistant to virus evolution. And so here we've looked at either neutralization or ADCC response to either the original Wuhan variant of SARS-CoV-2 or the Omicron variant. And this is all using hybrid serums, again, because people who've been vaccinated only and never infected don't give good enough responses for us really to see differences here. But you can see if we take these hybrid serums and we look at neutralization, there's a significant drop-off in neutralization activity against Omicron compared to Wuhan, about a 12-fold drop here. If we look at ADCC, there is a drop, I apologize, these are on different scale. This is a log scale, this is a linear scale. So there is a drop in ADCC activity between Wuhan and Omicron, but it's very, very small. So only about a 1.6-fold reduction in the ADCC response between these variants compared to a 12-fold reduction in the neutralization response. And so to conclude, if there's one sort of conclusion to come out of all our work together, it's that the antibody responses that are required for neutralization and for ADCC are very, very different. As I already said, really you want both of these responses to be present, so you have good activity against both cell-free and cell-associated virus. I still think if we want the best ADCC possible, we need to look to non-spike antigens. So all through A, membrane, and N really dominate the ADCC response following natural infection. And we've shown that antibodies targeting N can be effective in animal models, even in the absence of neutralization. And as I say, therefore, I think harnessing these additional antigens in vaccines and immunotherapeutics may be a way to improve efficacy. However, at the moment, what we have are spike vaccines only in the main. And spike, generally speaking, at least as far as vaccination, infection, or monoclonal antibodies, is a comparatively poor ADCC target. But we can improve that somewhat in the context of hybrid immunity. And this appears to be because infection leads to S2 focused responses, vaccination leads to S1 focused responses, and so in hybrid immunity you have this unique situation where you have both S1 and the S2 antibodies, and it's that polyclonal response that's needed for S-specific ADCC, which also, because it's so polyclonal, reduces the chances of variant escape. And so I think this could, at least in part, it's obviously not the whole story, but it could in part explain the superior protection that has been shown by many groups to be imparted by hybrid immunity. It also suggests that as we look towards future vaccines, lots of papers on neutralizing antibodies, you'll see comments on the fact that it could be worthwhile focusing your vaccines on the RBD to really drive the immune system to make neutralizing antibodies against neutralizing antibody targets. But I think our work suggests that could potentially be a mistake because you would miss out on the induction of potentially some ADCC responses. Lots of acknowledgements, of course. The first study I talked about was led by Kerry in Cardiff with collaborators in Cambridge, King's, and Guy's and St Thomas's. The mouse study with the N vaccination regime was done with Pablo and Justin at Northwestern University and UIC, and the hybrid immune study was done with Michael Grant and Kayla Holder at Memorial University. Thank you very much.